Good day. My name is Paul Wax. I'm the Executive Director of the American College of Medical Toxicology, and I'd like to welcome you all to our webinar series entitled The Medical and Public Health Considerations of COVID-19. Today's talk is going to be on the emergence of SARS-CoV-2 variants. Next slide. I'd like to thank our webinar series partners from around the world for helping to promote this webinar series, and they've all stayed with us now for over a year as we've continued this series, and many thanks for all the promotion. Next slide. This webinar will be recorded and will be available on the ACMT website within about 48 hours or so. Registrants will get an email about the exact location of the webinar, and feel free to watch it or pass it along to your colleagues. Next slide. We've also developed a series of FAQs for some of the webinars, and feel free to take a look at the FAQs we've developed also up on our website. Next slide. There will be a Q&A at the end of the webinar. This often goes on for 20 minutes or so. It's often a very interesting and robust part of the presentation, and feel free to type in your questions into the Q&A function, and we'll try to get to as many of the questions as we can. We often will go a few minutes beyond the top of the hour, again, to get to as many of the Q&A questions. Next slide. At this point, I'd like to introduce my co-moderator, who's Dr. Ziad Kazi. He's the Secretary Treasurer of ACMT and past president of the Middle East and North African Clinical Toxicology Association, and I'll be introducing our guest moderator for today.
Thank you, Dr. Wax. It's my pleasure to welcome Dr. Mark Zamet, who is a colleague, clinical toxicologist, board member of the European Association of Poison Centers and Clinical Toxicologists. He's also a faculty at the Department of Clinical Pharmacology and Therapeutics, Faculty of Medicine and Surgery in the University of Malta. In addition to his role in those two roles, he's also in the Ministry of Health of Malta and has been at the forefront of the Maltese response to COVID-19. And hopefully he will join us at future webinars as to provide us an update about the situation in Malta. Dr. Zamet, thank you so much. And Dr. Wax, back to you.
Thank you. Well, it's really my pleasure to introduce our speaker today. Dr. Anne Katrin Ullman is an associate professor of medicine in the Department of Medicine in the Division of Infectious Diseases, and is also the director of the Microbiome and Pathogen Genomics Core at Columbia University in New York City. She's really been at the cutting edge of research into these variants of SARS-CoV-2. She's done a lot of the work that's helped to identify new variants in New York City, and at this point is really one of the leading experts in the world to discuss this issue, which so many of us are eager to learn more about. So at this point, I'd like to turn this over to Dr. Ullman for her presentation. Thank you so much.
Thank you, Mark, and Ziad Paul, for the very kind introduction. Thank you to the American College of Toxicology for inviting me. I have received research funding from Merck and GSK in the past, unrelated to the current presentation. And I'm sorry, just having some issues with my slides. 1 second. There we go. So what I would like to discuss today is start off with an overview of the SARS-CoV-2 genome, and then discuss the current variants of concern, and as an example, present our work on the emergence of a novel variant from the New York perspective. And I think this can highlight a lot of the questions and difficulties that have arisen in this field. So, as we all know, the disease was first identified in December of 2019, and really at unprecedented speed. Within only 12 days or so, the first public genome sequence was available. Later in January of 2020, the Arctic Network published the first online protocol for genomic sequencing that really allowed for an unprecedented survey of viral sequences from all over the world. And I put these slides together just a couple of days ago, but we've now reached probably about 1 million of genome sequences that have been shared and are publicly available at the GISAID website. And I think this sort of data sharing highlights that a lot of the existing infrastructure was used and really leveraged to help respond to this unprecedented pandemic. This website, GISAID, was originally established to survey avian influenza spread. So how do we sequence the virus? Again, the Arctic protocol was actually developed to respond quickly to one of the Ebola outbreaks. And at its core principle, it uses tiling PCR to amplify the viral genome. And so the benefit of this approach is that it allows sequencing or amplification of genomic material from relatively sort of low input samples and clinical specimens. There are sort of multiple sequencing platforms out there, but the sort of two most commonly used are the Oxford Nanofold platform, which can be done in a handheld fashion or on a desktop computer. And one of the advantages is that it really provides rapid, near real-time sequencing. You can get the data as you sequence. And then, of course, an alternative is Illumina sequencing, which a lot of pathogen sequencing is being carried out with nowadays. So when we think about the SARS-CoV-2 genome, it's about 30,000 base pairs long. It encodes for 29 proteins. And as this is an RNA virus, we need a polymerase that sort of amplifies the RNA virus. Usually for viruses this size, you will find a lot of errors in this replication process. But SARS-CoV-2 has a unique feature, which is an intrinsic proofreading mechanism, which is thought to be due to the action of this non-structural protein 14. And because of that, it appears that the mutation rate is significantly lower than what would have been expected from other RNA viruses of this size. So early on, the notion was that because of this mechanism, we would actually see very few mutations compared to other RNA viruses, and that we could think about maybe about 30 mutations per year. So, when we sequence the viruses, we compare the sequences to a reference genome. In this case, most commonly, we still use the Wuhan virus as the reference sequence, and we determine where are the point mutations, and then we can use this information from the mutations to determine the relationship of viral samples to each other. And this is being done by phylogenetic analysis. And as you can see on this phylogenetic tree here, we have sort of different clusters that are falling out, and that represent how closely or how distantly samples are related. So if you have two identical viruses, the two dots would be very close together on the tree, and the more distant the sequences are, the longer the branches will be. So with that, I want to just address one of the really very confusing things about viral variants, which is how do we name them? And it's really quite confusing, as there are many different nomenclatures out there. So in general, we are really trying to name a clade, meaning a cluster of specific sequences that we can see on the phylogenetic tree, for example, the one I just demonstrated to you. The most commonly used mechanism that's being used now is the next strain nomenclature. And this uses the clade by year as well as a letter. And then the sort of second nomenclature is the pangolin COVID lineage assigner. And the way this works is that it gives an alphabetical prefix. So we're going from A to B, perhaps to C. And then we have a dot that identifies descendants of. And we have 3 hierarchical levels. So for example, we could have B, and then we have the first descendant, which is 1. And then B11 is the descendant of B1, etc. However, if you reach sort of too many dots and too many descendants, then we go to the next level, which would be C. And we are starting off, And we're starting off at C1. Sorry, it's a very responsive keyboard here. I'm just going to the right slide. Sorry about this. Stuck with, okay. I'm sorry, the slides are jumping, 1 second. Okay, so what we can then do is when we assign this sort of nomenclature, it's sort of what we are using to track variants in the global genomic databases. And what I'm showing you here is the evolution, or is the nomenclature using this next strain naming. We have the time scale at the bottom, and you see how over time, different lineages that are in this case defined by the year and then followed by the letter, and how they've changed over time. And if I can just draw your attention here to the 20I501 VY1, this is the next strain name for B117, or as in other naming, sometimes the geographic origin is also being used to name it. And so we sometimes call it the UK lineage as well. Obviously, there are a lot of limitations with using the geographic origin to refer to a lineage, because it can really be very misleading and may just be the first time where a sample was sequenced rather where it emerged. I will also say that there have been other attempts to break away from these naming schemes, and bird names have been proposed. But if you think of the viral diversity, this can get also very complicated very soon. So with that, I just want to sort of briefly discuss what drives variant emergence. So any living being accumulates mutations all the time. For viruses, we know that usually they accumulate relatively quickly. Many of these are synonymous or silent. That means they're not changing the information of the viral proteins, and they may not have a major effect. We do know, however, that there may be selective pressures for certain non-synonymous changes. And so as mutants emerge, they may not be beneficial, they may disappear in a population, or if they do provide an advantage, they may be maintained within this population. And so there are sort of three areas that we think about that may lead to selection of specific mutations. The first one is intrinsic fitness, and this could be fitness within the host during a persistent infection, or fitness during the host-to-host transmission process. So especially when a pathogen sort of enters into a new host, there may be a lot of selective pressure that needs to be overcome to establish the new infection. There may be direct interactions and alterations with host proteins and the host translational machinery. And then last but not least, certainly one of the selection pressures that is receiving a lot of attention right now is the escape from the adaptive immune response. So the first mutation that really gained a lot of attention was a substitution in the spike protein at position 614 from a D to a G. And it was actually the sort of first challenge to this notion that we weren't really expecting to see functionally relevant mutations in SARS-CoV-2. And as you can see on the top of this figure here, in the timeline, in sort of January, February, we had mainly virus circulating with the wild type. And then really in March, April, this G614 took over and essentially defined the North American surge of the viral infections. There was a lot of debate at the beginning if this was perhaps just in sort of a mutation that accumulated and perhaps wasn't necessarily of functional importance, but it's now been shown in a number of studies that it's clearly associated with higher viral load. And if you think of higher viral load being present, likely leading to higher transmission of the virus to new hosts. And as you will see, while of course there are mutations occurring across the SARS-CoV-2 genome, a lot of attention at the moment is being put on understanding the spike mutations. I will say that there have been a number of non-spike mutations described, for example, in the non-structural protein 12. And I think this is an area that will also gain more interest and attention, perhaps in the near future. But clearly right now, it appears that the spike as a protein that mediates a lot of the interaction with host receptors and also the host antibody response clearly is an area where a lot of the adaptation takes place. Now, I'm showing you here also a structural image of the spike protein, and it's really not just the sort of whole spike in general, but it's sort of the potential importance of just unique regions within spike. So this is in particular the receptor binding domain, where we know a lot of the antibody spike interactions take place, as well as the N-terminal domain. And then we have another of sort of other regions that likely are also hotspots for the accumulation of mutants. So with that, I want to start discussing some of the global variants of concern. And the first one that was recognized was B117. So the first isolate was detected in the south of England in September 2020, but it was really the dramatic increase in cases in this region in December of 2020 that led to the recognition that perhaps the virus itself might be contributing to this spike and surge in cases in England at the time. And I will say that I think the UK was really in a unique position to identify and track changes like this, that they had a really robust genomic surveillance system in place that I think has been quite unparalleled across the globe. So there are 8 mutations in the S protein compared to wild type, most notably this N501Y mutation. And it's been demonstrated that this leads to increased ACE2 receptor binding. There has been a lot of discussion on what the direct impact of clinical impact of this variant is. And so a number of themes have emerged. First off, as with the D614G, it's been demonstrated that patients who are infected with B117 on average have a higher viral load. And the estimate is that it's about 50% more transmissible than other variants. There also have been analyses and reports that it's associated with increased hospitalization and fatalities, but I will say that this has not been supported by all studies. And these are actually really sort of relatively difficult to discern events and outcomes in real time, as very often you actually still need a period of time to really be able to assess what the outcomes are. In terms of the sort of differences between these studies, I think they were all different in design, and I think a lot of the methodological differences explain the different findings, and I think it's something that still requires further study. Importantly, none of the studies have demonstrated that there is an impact of this variant on neutralization with monoclonal antibodies, and really just a minimal effect on neutralization by convalescence or post-vaccination sera. So it appears, therefore, that the big advantage, or the big fitness advantage of B117 is really its ability to generate a high viral load, to be more transmissible, and to bind to the ACE receptor. But we don't have a good indication, or we don't have any indication that it interferes with therapeutic or naturally acquired antibody responses. So this was sort of really the first lineage that garnered a lot of attention as being a variant that causes sort of more disease, and it's led to this terminology of the variant of concern. And the CDC has a few weeks ago issued sort of more detailed definitions of what this means. So there can be at least 2 of these criteria need to be fulfilled. It's the increase in transmissibility, the possibility of having more severe disease, and this could be manifested as either increased hospitalizations or an increased number of deaths. significant reduction in neutralization by antibodies generated during previous infection or vaccination, as well as the reduced effectiveness of treatments or vaccines, or also failure of current diagnostic tests. So we can see that B117 fulfills some, but not all of these criteria. So with that, there are a number of other classifications. The highest category that was issued is a variant of high consequence, but so far none of the lineages has made this classification. And essentially this would be that the virus is more transmissible, more severe disease, impaired diagnostic, impaired therapeutics, and really the anticipationists would require a different level of containment. So in addition to B117, we have B1351 and P1 as international variants of concern. And then just relatively recently, we have the B1427, 1429 from California that were added to this list. In addition to that, we have the classifications of the variant of interest. And the current examples are B1526, which I will be discussing in more detail, and then B1525 and P2. And the criteria for that are that there are concerns that these may fulfill, or that these are also associated with more transmission, more disease, but there are still sort of pending investigations to really ascertain what their clinical impact is. So B1351, also known as a South African lineage and sort of first described there, emerged in October 2020. Again, it has a number of signature mutations in the spike protein, and it shares the 501Y mutation with the B117 variant. In addition, though, there are two additional residues of particular concern, and I'll discuss them a little bit in more detail later, K417N and E484K, that appear to be interacting with the antibody response. But what really stood out, and I think really led to the identification that this variant lineage is of clinical concern, is that during a time when the Oxford vaccine trials were carried out, investigators in South Africa found that this vaccine was less effective. And even though the sample size was relatively small, this really raised significant concerns that the novel interventions that are being sort of prepared and the sort of vaccine preventive measures may not be as effective with this set of mutations. The third variant of concern sort of paints a different picture. So first, B117, we have the increased transmissibility. 351, the major concern was actually the failure of the vaccines. And then the P1 was described in Brazilian travelers to Japan, but really has gained a lot of attention in that it was first found to cause a significant number of infections in a region called Manaus. And from what we know is that when you look at the figures on the right here, in the sort of during the first spike, a lot of infections occurred. And the thought is that about 50% of the population were infected. And despite this background of having a very high seroprevalence in the sort of fall and winter of 2020, a second large wave of infections hit despite this high seroprevalence. Again, we have multiple spike mutations, and they do overlap with those that have been described from B1351, but actually still have additional changes in spike. So P1 has now accounted for a vast majority of the cases in Brazil. And as I'm sure you're all aware, the surge in Brazil has been high and ongoing for the past few weeks. So while the epidemiology really helps identify a lot of these variants and lineages of concern, we sort of need functional data to be able to look a little bit more into what the sort of clinical impact might be. And I'm showing you here just a panel from work conducted in the laboratory of my colleague David Ho at Columbia, where he and his team investigated the neutralization of B1351 by monoclonal antibodies. And so what this team did, they investigated the wild type and then generated a pseudovirus, which is a virus that only sort of has the spike protein. They recreated the sort of combination of all of the spike mutation and then sort of generated single mutants. And what I'm showing you is that the neutralization by the monoclonal antibodies with EUA, under EUA, were all reduced, significantly reduced in the wild type, as well as in the virus that had the combination of these mutations. And then though, when you look down and look at which one are the key sort of residues that are responsible for it, it's the E484K mutation that stands out and seems to be responsible for most of the effect that's being observed here. In addition to the decreased neutralization by monoclonal antibodies, the team also observed decreased neutralization by convalescent plasma and by post-vaccinee sera. So what this brings up is really that while a lot of classification takes place on the level of variants of concern, should we really be thinking, at least in part, about mutations of concern? And certainly E484K seems to be a prime candidate for this. So it's present in 2 of the 3 major variants of concern. But when we look in the public databases, we can find that it's been described in at least 100 SARS-CoV-2 lineages. And to give a ballpark, I think at the moment with the various classification schemes, they're perhaps up to 800. But most of the data really so far we only have from the 2 sort of variants of concern. So with that in mind, we sort of decided that we wanted to really investigate how widespread these were in our catchment area in northern Manhattan in New York City. And to be able to do this quickly, David and one of the colleagues in his group developed a rapid screening assay. So it's a very simple RT-PCR assay of an inactivated sample, and that allows us to just get this genotypic information on residues. We initially started off looking at the E484K mutation as well as the N501Y. And what we found when we sort of started looking at this right around December, January, when the international reports came out, was that very early on, there were sort of very low levels of positives for the E484K mutation shown in the purple here. But as you can see over time, really reaching into sort of the late January, we see a steady increase up to 25% of all our isolates that were sort of identified as being positive in the clinical microbiology lab. In green, I have the 501Y, which is the signature mutation of the B117 lineage, and actually at this time there are very few cases with this. So our expectation was that perhaps most of these isolates would be consistent with the international lineages that were described, and to sort of investigate this in more detail, the postdoc in my group, Maidani Anavachala, sequenced these mutant samples that we identified, as well as matched random samples that were negative in the mutation screen. And if I can just sort of take you through this relatively busy phylogenetic tree. So on the left here, I'm showing you sort of the different clades as I demonstrated earlier. If you have small branches, they're very closely related to each other. And then up on top, I have the spike protein and the many different mutations and residues that are changed in the spike protein. And to our surprise, we found that really the majority of samples clustered together in a novel lineage, which is the B1526. We had a couple of the B1351 and then the 117 isolates as well, as was expected from the PCR screen. So in addition, though, it also became apparent that there were a couple still very closely related subclades, which you can see here, which do not have the change at 484K, but instead actually have an S477N mutation, as well as more recently an L452R mutation. So it suggests that we have the accumulation of the E484K mutation in a lineage that, when comparing to the public databases, appears to be originating in the New York area, and then we have sort of closely related sublineages. So when we look at this in a little bit more detail and sort of try to reconstruct what were the key steps that led to the emergence of these lineages, we can sort of reconstruct the evolutionary history. We see that it's one of the mutations in the N-terminal domain that arose first, and then we sort of have these different branching points, the E484K branch, in a way we've selected for this, right, being interested in this residue, as well as this other somewhat unexpected mutation, S477N. And so if you're just looking again at the structure of the S protein, we see that those two both are in the receptor binding domain. And then we have the sort of a little bit also present in all of them is this A701 mutation, A701B, which is next to the furin cleavage site, that potentially may play a role in transmissibility as well. So to then ask the question, well, how does this translate into functional changes? Pei Feng and David's group reconfigured the pseudoviruses, and let me take you sort of through his essays here. So the first is we have the wild type and an S477N pseudovirus, and as you can see here, the neutralization with monoclonal antibodies currently approved or in development in the sort of broken up lines, there's really no difference if you just change that one residue. Likewise, if we add the additional mutations that are present in this 477 lineage, we again don't see any differences. In contrast, and not unexpected, if we introduce the E484K mutation, we see the significant impact on the neutralization of some of the monoclonal antibodies, not all. And then if we add the additional mutations that are present in the 5 to 6 lineage, we see a couple of additional changes and reduction in the neutralization by monoclonal antibodies. At the bottom panel, and I'm sorry that this was cut off here, but we are comparing convalescent plasma and then the post-vaccinee sera. And this is in this case only looking at the New York Delta 5. So this is the construct that has the 484K plus the additional mutations in the spike protein. And what you can see is that there is a reduction in neutralization, but it's really relatively minor. So for the post-vaccinee sera, we are in the 3.3 to 3.6 range. And in the convalescent plasma, we are sort of a 4.1-fold decrease. So what have we learned so far about the clinical findings? And this is really an analysis that's very much in progress. This is sort of our first set of analyses where we have 150 patients with the E484K mutation compared to those that were typed as wild type. And we need to keep in mind that the majority of the patients that we are seeing are being admitted or are coming to the emergency room. So it's not a population that sort of represents all of the levels of care. And really so far, there are relatively few clinical differences. So really none in the sort of common demographics. We have a little bit of a difference in terms of comorbidity just by diabetes being more prevalent in the patients with a mutation. Significance of this is not fully clear yet. I should really highlight that we have a higher proportion of patients who are still admitted with the mutation compared to the wild type. So a lot of our outcomes data is still pending. But I do want to point out that, as has been described with some of the other sort of variants, we see that the cycle threshold value, which is sort of the PCR test that's a little bit a surrogate for the viral load, is lower, meaning reflecting a higher viral load in those with a mutation compared to the wild type. Now, when we look at the geographic distribution of these mutant cases, so on the left, I'm showing you the sort of zip codes and the catchment area of all patients who present to our hospital. And on the right, we have the ones reflecting patients with this particular lineage. You can see the sort of gradual emergence and a pattern that doesn't fully reflect the overall catchment area of the patient, sort of highlighting potential initial local expansion. And sort of subsequent studies and analyses now demonstrate that our data are also replicated by other academic centers in the city, and certainly there has been sort of an increase of the cases across Manhattan and the 5 boroughs as well as the suburbs. Now, all of this has been sort of taking place in the context of case numbers remaining relatively flat. So while I pointed out that with B117 in the UK, in a way, the reason I think it was found quickly was a steep increase in the number of cases. We haven't seen the sort of same pattern. This hasn't happened in the context of a rapid increase in cases. Rather, we've pretty much had a very flat curve since about the sort of end of December holiday spike that's been seen in many parts of the country. And so the current thinking is that, of course, in December is also the time when the vaccines started rolling out. And so that we are in a situation where we have, hopefully are seeing the initial positive effects of the vaccine at the time when we have the emergence of a variant that perhaps has some advantages. So I do want to just point out that when we compare, while it's sort of been great to have these genomic databases, that when we compare data, there may always be caveats in those databases. For our own data set, we can say we really typed and sequenced all of the samples that were available, but what's in public databases may be slightly different. And so one of the caveats is that actually with most of our sequencing, the best quality data only come for samples that have this cycle threshold value of less than 30. However, you might have a positive test at 40 or 45. And this does mean that with our current sequencing, a large viral reservoir is not being monitored. And there are also other biases in the sample selection. For example, with the B117 lineage, there's a deletion in the spike protein, and it leads to an S gene dropout in one of the diagnostic tests. And certainly at the beginning of the year in the US, there was preferential sequencing of these type of samples. And so it may have actually led to a little bit of an overrepresentation of B117 in public databases. With that in mind, however, if we compare growth dynamics using the public data sets and compare the B1526 to B117 in our own catchment area, we see that 1526 significantly outpaces 117, certainly for the beginning of the year. And then we are looking at a doubling time of about 15 days. And when we expand this analysis with all of these caveats that I mentioned, we see that the sort of growth rate for 117 as well as 1526 is currently on par in the New York State area. And with that, I want to say, while we've seen the initial emergence of this variant in the New York area, if we now sort of just track what's been going on over the past sort of 3 months or so in the US, we see that there are significant case number reported from many other parts of the country, and also there are increasingly these sequences described from international sites in the databases. So where are we? What should we expect next? And so I'm just highlighting our data again, sort of summarizing the genomic data, the phylogenetic tree, each of these colors representing a different sort of lineage. And if we sort of sum it up in our own data set, as well as in the New York City data, about 80% of all of the samples appear to be accounted for by sort of variants of interest or concern. I also want to point out that in the orange here, we have in the 5 to 6 lineage a mutation at the L452R position. And this is actually what's also been present in the California variant, or the 1427, 1429. So while our focus has been on 484K, clearly L452R appears to be another residue that perhaps keeps mutating and keeps popping up in different settings. And this, as I had pointed out earlier, Lineage was recently added to the list of the variants of concerns. And it's been demonstrated, again, the impact on the neutralization by some antibodies, and then the moderate reduction in neutralization using convalescent post-vaccination sera. And it appears to be about 20% increased transmissibility. So with that, there are still many ongoing clinical questions. And the first one, I think, really relates to transmissibility and how we're going to measure this. So, right, we are in a situation now where it appears we have a handful of variants that appear to be competing and that account for most of the cases. So as we are examining this, it's really an analysis of competition between variants. There's still questions about the severity of disease, and also, again, how we're going to measure it, taking into account some of the novel or neurotherapeutic interventions that we have. And I think the question most of us are really interested in most is, are any of these mutations or lineages really able to cause infections in previously infected and or vaccinated individuals? And is that sort of really going to be the next area of concern? So with that, to summarize, SARS-CoV-2 is continuing to evolve. We have novel variant lineages, but we also have specific mutations that perhaps independent of the lineage background may be contributing to a lot of disease and difficulties that we are still encountering. In the spike protein, E484K appears to be occurring in many different lineages, really indicating its potential functional importance. There may be some other sort of factors in, for example, the backbone of 526 that contribute to the recent success of this lineage. And I think the question really is now, should we be monitoring lineages versus mutations? And also, as the lineages have emerged, they will continue to evolve, either to be more fit or perhaps also at some point to really run their course and not cause as significant disease. Concerns are that some of the monoclonal antibodies are not as effective anymore, and there have been some changes in the treatment strategies. And I think with all of these concerns, the key message is really we need to drive the numbers down, and the importance of vaccinations and other protective measures, I think, has never been bigger than now. So with that, I really just want to acknowledge the many people who have made this work possible, in particular in my lab. My postdoc made me, the collaboration with the David Ho lab, the many others in the division and the university who have helped. And with that, thank you for your attention, and I look forward to our panel discussion.
Thank you very much, Professor Uleman. An excellent presentation. I would like to ask you one question. I would appreciate your insight and feedback also with regards to a study which was in the news last week, an Israeli study, which basically concludes that the B.1.351, the South African strain, is 8 times better able to break through vaccine protection when compared to other variants. So I would like your insight on this. And also, what relevance do you think such conclusions would result with regards to, for example, relaxation of measures?
Sure. So, I think it's always difficult to, how do you weigh fold changes? So we clearly know that with the, the concern with the 1351 has been that the Oxford vaccine is not as effective. And the concerns have been that if in an area this is the predominant lineage, that this should not be the vaccine that's being administered. There's been some debate if there's also a reduced efficacy with, for example, the Pfizer vaccine. So I think if in a sort of given geographic area, if you're in a position where there is, there is the ability to use other vaccines, it's, we should use what appears to be most effective. But I think it really highlights that the overall, the global burden of the virus has to be reduced to really reduce the impact of these mutations or variants.
Another question. Also, what is the criteria for variants of concern becoming variants of high consequence?
Mm-hmm. So the CDC has only sort of put up these sort of definitions, and none of the variants have made it to that level. My understanding is that you would really see impairment across all groups. So increased transmission, more disease severity, ineffective therapeutic measures, and also ineffective preventive measures. So really fulfilling all of the criteria across the board. Honestly, hopefully we will not get to that level.
Another question is also, is there sequencing being done on specimens from people suffering breakthrough infections? Mm-hmm. Sequencing in a systematic way?
Yeah. So the CDC is collecting information on these cases, and I can say, for example, for our institution, we are, this is an area for us of particular interest, and we are trying to prioritize sequencing of these samples. I will say that the breakthrough infections post-vaccine that we've seen, there have been relatively few, which is good, and often though, I think sometimes the viral load in those post-vaccine infections is low. And as I said before, technically, sort of, you get best sequencing results with a CT value of less than 30. You can actually still get decent information until 35. Anything that's in this 35 to 40 range is really much more difficult. So that means that I think for some of these samples, it's just going to be very difficult to type them. But I think that's where we really need to focus on a little bit to understand, are there specific mutations that occur in those isolates. I would say the short readout right now from what we know is it doesn't appear that there is a major, at least in the US, sort of one single lineage that accounts for those infections.
Another question, a very interesting one. Is there the possibility of polyvalent versus monovalent vaccines as a multiplicative strategy in addressing variants? What do you think?
Yeah, I think that's an area of great interest and might be the way forward. And I would say at the moment also, I think there's a lot of interest in, for example, studying the natural response of individuals who, let's say, previously had an infection and are now vaccinated. So is their immune response going to be different compared to those who are just vaccinated, or other permutations of this?
So Professor Katzi?
Yes, thank you, Dr. Zamet. I'll have a couple of questions from the audience. Are the mutations unusual for antisense single-strand RNA viruses? Are all mutations expressed, or are there mechanisms such as folding or methylation that block mutation expression?
So, the mutations are unusual in that they don't occur as frequently as would be expected for this type of virus. And it appears to be related to this proofreading exonuclease that's part of this NSP14. So it's just that there is, a better proofreading mechanism sort of present. And so we see a lower number. In terms of, location or other changes, I think there's still a lot that we need to analyze and learn.
Interesting. And then as you may have seen, the surge in Michigan,
mm-hmm.
Have your colleagues been working in Michigan on identifying potential lineages or mutations that are driving that surge?
Mm-hmm. So as far as I know, the vast majority of these is really due to the B117. And it's, it's sort of, I think it highlights a lot of the things that perhaps we don't understand. I don't think it's really clear why do we see this surge right now in Michigan, for example. So the CDC and local labs have been monitoring the presence of variants across the country. And actually the state that had most B117 was Florida. So everyone has been watching Florida, sort of waiting for the cases to go up. So the notion is that if you have a lineage with sort of higher transmissibility, once you reach 50%, you're expected to see this sort of surge. But actually, I mean, Florida numbers maybe are bouncing around, going up a little bit, but they didn't have the surge. So why did it happen in Michigan now? I don't have a great explanation. We know from the sequences there appear to be additional mutations that are popping up. As I said before, right, even when you're in a lineage, it continues to evolve. I don't think we know if any of those may play a role in this as well.
Interesting. Now, commonly, I have family friends that ask me about, what they're able to measure on a swab for the viral count, or for example, the level of antibodies that they have in these commercial labs. Can you comment on those being, not an accurate or not a clinically useful estimate or value?
So the first part is right. What do we get from the virus, the sort of clinical information? So the clinical test by the PCR is the RT-PCR, and so you have the cycle threshold value. And there have been a number of studies indicating that sort of on a, in a larger population, a lower cycle threshold, meaning a higher viral load, is associated with worse outcomes. So not everyone who has high viral load will have bad outcome, but sort of on average, you see that. There has been a lot of debate if the cycle threshold value should be ordered in the medical chart. So is it sort of meaningful information to communicate to providers and or the patient? Certainly this notion, this association has been shown across many different platforms, but you really need, a standard curve. You need sort of robust measures, and there's, I think, have just been some concerns that it's not always accurate. So I think most institutions probably do not communicate this. The viral load certainly has implications for transmission. So there have been studies trying to associate this cycle threshold value with sort of viable virus, which would indicate the possibility to transmit. And the thought is that perhaps around 34, so there's relatively little, but it's only this one moment in time, and we don't know if somebody's going up or down, so it's probably also not that meaningful in that setting. So I think we need, this needs to be fine-tuned a little bit more. In terms of the antibodies, I think that's sort of the new evolving area in question, right? So now post-vaccination, should we be measuring antibodies? If so, which ones? Do the levels indicate anything? And right now, I think, the current understanding is that we really don't know yet what those levels mean. So as of now, there is really no recommendation to check post-vaccination, let's say, spike protein antibodies or even neutralizing antibodies. And this may change, but right now I would not recommend it.
But as far as our public health measures, the urge to vaccinate people rapidly, can you comment on that? Can you make a statement about that? How important that is for our country to get vaccinated, and how would that affect our response to these mutations?
So, I think the goal really should be to vaccinate as many people as quickly as possible. And I think we have now sort of at least 2 countries with really good data, real life data to support this, the UK and Israel. And so clearly in both countries, I think it's been demonstrated how this large vaccination effort can drive down case numbers. And I think that has to be our goal. I think, when we think sort of, and this is sort of more speculative, but if we think of sort of a pathogen reservoir, right, the larger the reservoir, the more opportunities for mutations, and right, so it's random changes all the time, and you may just end up with this perfect that may may persist and may sort of go on to cause more disease. So as much as we can drive this down, I think the implications are, more people are protected, but also if there is transmission, most likely it's lower viral load. So I think it's really hugely important.
I'll ask one last question and turn it over to Dr. Wax. Considering the variants we have in the US and the mutations you so well described, do you think using the Pfizer vaccine, the Moderna vaccine, and the Janssen vaccine that is currently on hold but may come back, do you think we have, how much time do you think we have to, where these vaccines are still going to be effective before the mutants win, if I may say?
Sure.
And thank you.
Sure. So I don't think we have any true indications that with any of those variant lineages that we have significant vaccine failure. So it's not that, we are in a situation where we only have, let's say, an AstraZeneca vaccine and all of the cases are due to be B1351. So I think that's an encouraging situation. But of course, we don't know what else is evolving. So again, I think just driving the numbers down as much as possible, and obviously locally, but also globally. So while we see decreases in numbers, and that's encouraging, worldwide, the numbers over the past 2 or 3 weeks have not been positive.
It's my understanding that there have been a very small number of people that, despite having completed their vaccination series, that they've been infected with COVID, and in fact, some of them have been hospitalized. Could you describe perhaps those sorts of patients that have been infected with COVID despite the vaccination, and also whether exposure to a variant may have played a role in their infection?
Sure, that's a great question, really one of the hot topics right now. So I can just speak from our own experience at our hospital. And so yes, we do see some infections after vaccination. And in a way, that was to be expected, right? Because we know the efficacy, even in the trials, wasn't 100%, it sort of was in the 95% range. What's very difficult to sort of judge is the numbers that we are seeing, are they sort of out of the range of what should be expected? And so we have to really think about the denominator, how many people were vaccinated, and the people we see in the clinic or in the hospital is just this sort of small portion of this, and from all we know, a really huge denominator. So in the New York area, right, millions of people have been vaccinated. So the way we've been analyzing the data and really looking at them constantly is that we are interested to track. So full protection is defined as 2 weeks post the second vaccine for those with the two-shot regimen. So we look at those, but then we also do look at the stages before. So just prior to the second vaccine, or 1 week or 2 weeks after that first. And so there have been a handful fully vaccinated 2 weeks out. I would say that the majority of the cases I've seen are in people with significant underlying health conditions and immunosuppressive treatment. So I think special patient populations that, may have been sick again because of underlying conditions, or perhaps didn't have as robust of an immune response. But I really want to stress that those numbers are very small from all we know. We are sequencing those isolates. I know the CDC is collecting data, local health departments are collecting data. And at the moment, there is really no indication that it's specific variants or mutations that account for these cases, but it's really all evolving in real time. I do think that potentially we also may be having a little bit of a problem with the psychological impact and the excitement about being vaccinated, and especially even after that sort of first vaccine, and how are people changing their behavior and perhaps already dodging some of the caution and sort of taking a bit more risk than they might have done before, which may account for some of those who are infected between the first and the second shot.
Okay, great, thank you. Another question is, Is there any specific variation in the response to viral loads with the B117 or other variants to therapeutics such as remdesivir or steroids?
Mm-hmm. So I'm not aware of any differences, but I would say because routinely, right, when we give remdesivir, there isn't necessarily a routine sort of follow-up on the viral load. I think we may also just not have enough data on this. But again, if there's a higher sort of bioburden, certainly the concern for potential accumulation of resistance is certainly higher and is a concern. I wouldn't be so concerned about the steroid response just because it's more geared towards the host. So I'm not aware that there are differences there. But certainly with remdesivir, I think that's a good thought and potential valid concern.
Okay, thank you. I'd like to just turn it back over to Dr. Zamet and Kazi for any last questions.
Yes, another question is with regards to the Tanzanian variant, which is interesting because it is evolving not from the B lineage, but from the A lineage. Mm-hmm. And also, attendees are asking about this variant and whether we should be concerned, whether we should be expecting it. Any feedback, please?
So it's a great question. I think it's always, when these pop up, it's always difficult to predict. For example, relatively speaking, I would say we've been very concerned about the B1351. And actually, while there have been cases described, for example, in the US, we've seen relatively little to date. There's perhaps a notion that recently the cases of P1 have gone up a little bit. We're still in the low percentage range, but the fold changes are higher. It's that geographic proximity that perhaps plays into this. Certainly, it appears now there is some evidence for local transmission. So when we have a variant or emerge in a specific geographic area, there may be this possibility there is a selective advantage and so a lot of potential for spread and seeding sort of larger outbreaks. There may also be local founder effects. So unfortunately, only time will tell how this plays out. But, maybe to just add, I think what's changed to maybe 4 or 5 months ago is that there we had a sort of variant emerge against most likely a lot of wild type virus. What we are seeing now is we have several variants, and I hope I was really able to get across that each of these, if you think of the 3 variants of concern right now, they each have slightly different advantages from what we know. And so in a way, it's a competition of these different advantages. And perhaps as novel variants emerge, they will find sort of other gaps in this. But it's almost a competition of those rather than against the wild type.
Thank you. Another question is also with regard to the difference between the spread of the UK, the B117 variant in Europe compared to the US. Mm-hmm. In the sense that in many European countries, it would seem that the B117 has caused increased mortality, significant decrease in the age of hospitalized patients in the 3rd and 4th waves. Could you perhaps comment on the differences between the situation in Europe and the USA with regards to this variant?
Yeah, so it's a great question, and I don't have a straightforward answer. I think one thing that I find sort of personally interesting and impressive about the pandemic is really these waves that we are seeing. And I don't think we always have a good explanation for them. I don't think we always know why it's going up and also why it's coming down. And so I think in Europe, right, we had this rapid increase in the UK at a time of a lot of holiday travel. Then it came down. And then already in other European countries, we had a high threshold, and then it sort of added and swept through. In the US early on, we've seen the cases, but there hasn't necessarily been so much going on. But now we sort of are reaching increases in some of the geographic areas. So I think in terms of mortality and outcomes, one thing I do wonder is, right, as a surge happens and more cases occur, I think there are two factors at play. One is higher viral load and more transmission. And so when transmission takes place, the viral burden is higher, and that may be a predictor of worse outcomes. I think that's one. The other is that once we have a huge influx of patients, it's really stretching the healthcare system and the ability to provide care. So, right, if you're in a situation where you have your overflow ICU in a gift shop, it's just probably, despite all best efforts, just not optimal. So I think it's really a multifaceted situation. But why do we sometimes have really this large spread in certain areas and not others? I think there's a lot to be learned.
Good. And another question is also with regards to the rise of variants of concern being possibly camouflaged by falling rate of infection overall. So potentially giving a false sense of security and leading to mature relaxation of restrictions. What would you say about that?
Yeah, I think that's certainly possible. And I think, it sort of has to be, we can only really figure it out sort of by very multidisciplinary approaches, right? Certainly the genomic sequencing to understand what's present, the epidemiology, understanding are the case rates higher, outcomes, but outcome studies take time, right? It's just you can't predict what's going to happen. And then, functional assays. So it's certain these variants emerge and remain undetected because the case numbers are not going up. And I think that's why, and CDC and NIH, I think, are very much focused on this now to really have early detection systems, identify which ones are the candidates. And it's okay if you look at candidates that perhaps in the end are not the ones that sort of rise, but the more we understand of what's critical, I think the better we can predict.
Okay, I just have
Thank you very much.
concluding questions. Dr. Kazi has a question. I have two final questions.
Thank you. Just a quick question for me. Has anyone studied the issue with the vaccinated people and whether they are carrying the virus and are asymptomatic? Do you have any information on that? And thank you.
Sure. So, as far as I know, in the vaccine trials, right, the outcomes was very much driven by reporting of symptoms. So I don't think from those trials we know much. I know that there are sort of ongoing studies in many of the sort of, I think especially healthcare worker cohorts who have sort of predefined sampling. And it's certainly really a very important area right now, right? Because that could be really an important reservoir, somebody who sort of is asymptomatic, carries it and can still pass it on. But I would say the data we have, though, do not suggest that this is a large proportion. So, I think it comes down to very few from what we know right now.
Thank you. Back to you, Dr. Wax.
Okay, and final two questions. Have there been any studies showing differences in the clinical presentation of any of the variants of concern?
So the short answer is not that I'm aware of. I think they may very well be, but I think the clinical spectrum, is just really very large, and I think there are just such strong differences. But I would expect that these are, findings that hopefully we will still sort of figure out in the weeks and months to come. I think the difficult part is, right, you could only really capture this with a pre-existing protocol where you're really classifying symptoms. And I think just as part of routine medical care, that's just often not possible. But, at least, for example, at our hospital, for a subset of patients, we're really trying to capture it. But there's no indication at the moment to say, well, this variant leads to more, let's say, cardiac involvement or more of the sort of clot burden. But I think, there are studies ongoing to really get at this. Again, it can be difficult because, right, some of the people who are diagnosed, it's so difficult to really get the genomic information because it's just so little virus, and that also complicates this sort of genotype predicting clinical presentation analysis.
Thank you. And final question, are there any differences by demographics such as race and ethnicity vis-a-vis the prevalence of the different strains?
So when we look at our data set, we do see some differences, just reflecting that a large part of our patient population comes from the sort of northern Manhattan and Bronx area. But you know, as for example, if you just think about 5 to 6 as it's been sort of spreading through the city, there's no indication that there are differences in that respect. And I think for any of the lineages we've sort of seen now, they can spread in different areas. You know, caveat being you have high viral load, you have a lot of transmission areas with crowding. You know, may of course be hit more. So I think these are difficult analyses as well.
Great. Well, you know, thank you very much, Dr. Ullman, for really an outstanding presentation. It was really everything we'd hoped for in terms of you providing us with the latest science and data, and I very much appreciate that you took the time to address us today. If you could go to the next slide. Our webinar series will be continuing next Wednesday. As it so happens, we had lined up our speaker from J&J several weeks ago before the pause to talk about the Janssen vaccine. But as of yesterday, with recent communication with her, she's still saying that you know, she's planning to give the talk next Wednesday at 3 p.m. Eastern. So it should be a very interesting presentation. Of course, there's been a lot of attention to the Janssen vaccine over the last few days. So we'll let everyone know if we have to postpone this talk, but at this point, it's still planned for next Wednesday. Next slide. Finally, all of our past webinars and upcoming webinars are in our prospectus, which you can get on our website at the URL on the screen. Again, I'd like to thank our speaker. I'd like to thank our guest moderator for an excellent Q&A session, and everyone should have a good day and stay safe.